All right, picking up where we left off with our P generation with the mamas over here, or the females, which have yellow bodies, short or miniature wings, and white eyes, and the males that have black bodies, regular wings, normal wing length, and red eyes. The F1 generation, the females, you'll see, look different from the originals, and so do the males. In fact, you have the reverse trend. When you get to the F2 generation, where you let the offspring interbreed, so remember now flies can't self-fertilize, so in the F2 generation here, what you're looking at is the offspring from the parents, the F1s, crossbreeding. Of course, you end up with females and males, but here you're going to end up with gray body, red eyes, long wings, or black body. I called it black, but they're calling it gray here. Yellow body, white eyes, miniature wings. So what you're looking at here are different combinations. These two are the original combinations, or the parental phenotypes. Basically, what you're looking at is that the parental phenotypes have the largest proportion of individuals. So females and males, you have about 700 that look like this, 750 that look like this, but then you very rarely get these gray body, white eyes, long wings, one female, 0 males. You only get one of those. Over here, yellow body, red eyes, miniature wings, you never get. Yellow body, red eyes, long wings, you rarely get. And the gray body, white eyes, miniature wings, you rarely get too. A little bit more commonly, but not by much. And so what that means is that all of these genes, all 3 genes, are located on the X chromosome. And because of that, they're transmitted together as a unit, which we call a linkage group. So although the flies with the parental traits were the most prevalent, the most common, you occasionally got recombinants. And what that means is that because you got very few recombinants, those genes are really close together. In other words, crossing over doesn't separate them and give you new combinations. So if you look at the analysis by gene pairs, instead of saying body color, eye color, wing length, when you look at gray body, red eyes, you got a bunch of those. When you look at yellow body, red eyes, you got very few of those. You look at yellow body, white eyes, you got a lot of these. Why was that the case? Well, remember, these were the parental genotype, or phenotypes, genotypes as well. The new combinations that you're getting, the rare ones right here, these are all recombinants, okay? Very uncommon. These are also recombinants, but they're a little bit more common. The red eyes, miniature wings, white eyes, normal wings. In other words, you can mix that up more easily. But body color and eye color, you can't really mix that or separate too well. So the rare recombinants, the most rare recombinants, oops, sorry, wrong thing going on here. I'll just have to cross that out. Here we go. The rare recombinants, these guys are the ones that are the genes that are closest together. In other words, body color and eye color are super duper close together. And so what that led to was additional experimentation and observation, where groups of Morgan students observed what was happening during meiosis, and they actually saw chiasmata, and they proposed at that point, that's what's mixing these things up. And so crossing over between homologous chromosomes resulted in generating recombinants. Now, how do they know this? It turns out that the Y chromosome has a weird shape. It has this little kind of hook shape. It looks kind of like a candy cane, a blue candy cane. The female X chromosomes are very long, so they're easy to kind of distinguish from the Y chromosome. It turns out if you have, in the F1 generation here, you have a female that is Y plus Y, W plus W, and this individual, the male, has Y and W. Then you can predict what male and female genotypes you're going to end up with, and you can predict the phenotypes as well. So with no crossing over, you would expect to get all gray body, red eyes, yellow body, white eyes. With crossing over, in other words, generating recombinants, you should get gray body, white eyes, yellow body, red eyes, which they did. 17 of these guys and 12 of these guys, or these guys and girls. So what this tells you is that all these genes are located on the X chromosome. when crossing over occurs in meiosis 1, that can mix up genes that are far apart on the chromosome. The genes that are real close together on the chromosome are difficult to separate from one another, which makes sense. If you have crossing over happening at the tips, crossing this part over with another part over here would definitely give you a new recombinant, or much frequently a recombinant, compared to two things that are close together. And so in this figure here, here's no crossing over, okay, and here's where you get recombination again, down low. And this recombination is going to give you a change in wing length versus eye color. So eye color is down here. So what you're looking at are a bunch of slides showing you different possibilities for crossing over, and basically showing you how often or how hard it would be to separate body color, eye color, and wing length. So it's very rare sometimes to separate these things. And basically it came down to needing a mathematical or statistical test to determine whether or not these recombinants were random. So guess what? Ta-da! Here we are, back to chi-square analysis, which you guys don't get to do in this class. Sorry to disappoint you. Of course, we're not doing this because it's not required for introductory genetics. You need to know what it is. You need to know why you use it. You need a statistical test to verify that these combinations didn't arise randomly. So this series of steps is showing you the hypotheses that you would be doing for the calculation. This would be showing you this particular Punnett square. This is basically a dihybrid cross. However, on this particular case, since you only have X and Y, only the X's participate in transmitting these traits. So you don't have to draw out 16 quadrants. You only need 2 because you're looking at X and Y. Here's the math. Apply the formula. Step 4. Okay, you can go on ahead and if you love statistics, go on ahead and wrap your mind around that. That's just showing you kind of the process that Morgan's group went through to determine whether or not crossing over gave you the recombinants that were, or that you could predict the recombinants. Now, direct observation was harder to do. Yes, Morgan's group saw chiasmata occurring in homologous chromatid pairs, all right? However, what was difficult was that X chromosomes are really long, and chromosome 1 in flies are also really long. So it wasn't until Barbara Mcclintock and Harriet Crichton did work on corn. Now, Barbara Mcclintock is very famous for doing work in what we call Indian corn. It's the colored corn that you see coming out a lot of times during Halloween and Thanksgiving. And the corn can be purple and yellow, or sometimes it's that real pretty red color with yellow grains as well. Basically what's happening in Native American corn is that the corn itself, the strain of corn that she was using had an odd chromosome number 9. Chromosome number 9 has 2 physical abnormalities, or 2 physical options, I guess you could say. The normal chromosome number 9 itself is just a specific length with, sorry, let me use my pen, that was my mouse, with a slightly offset centromere. The abnormal chromosome 9 has a piece of chromosome 8 that becomes translocated onto 9 during meiosis. Now, don't worry about the translocation right now. We'll talk about that later in this class. But the abnormal chromosome 9 also has this weird knob on the end. So it turns out that you can look at the differences in individual cells of a corn kernel and look at the chromosome 9. Now, something I want to stop for a minute here to point out to you guys. When you're looking at a piece of corn, here's Dr. Val's drawing of a piece of corn. That piece of corn is made up of many individual little cells, and that's what this dot represents. Each little cell, if you look at the individual cell, now let's try to blow up the individual cell. The cell itself has a nucleus and, mitochondria, chloroplast. Actually, these ones don't have chloroplast, I don't think. They have starch granules, but they also have the potential to have pigment. Okay, and that's what these little dots are, pigment. Here's the nucleus. So this is a corn cell. And each corn kernel, or corn seed, is made up of millions, hundreds of thousands, if not millions of these little cells. So what you're looking at is within each cell, within each nucleus, individual chromosomes. So this is one of the things that you don't always think about until you take a college level biology class, is that each individual corn seed is made up of cells, and some of these cells will have pigment and some won't. So let's say these little circles are cells that do not produce pigment. If they don't produce pigment, they might be missing the allele to do so. The ones who produce pigment are going to be solid colored. And so in the Native American corn that we see, you will see some pieces of corn that have a variegated kind of stripy look to them. Usually it's the purple corn. So here's the little V, that thing down here, and then you'll see spots on them. Okay, so Mcclintock knew that there were two genes that gave rise to that purple color. And we're going to just deal with the purple color right now. There's C for colored, which is purple. There is also a variant that's red. And then there's colorless, which is basically like a yellow color. Now, it turns out that each seed can also be puffy or it can be constricted. So a regular piece of corn kind of looks, round like that. The constricted one or the wrinkled one looks kind of, that's really terrible. Oh my gosh, Dr. Val, take an art class already. All right, I'm going to get rid of all that. That's a mess. Basically, you have a regular looking piece of corn and then you have a wrinkled one. I actually have a picture. I just thought I'd try to draw it. It turns out that when you look at the parental chromosomes, remember chromosome number 9, the normal one in this particular case is carrying the recessive allele for color. So C equals color. Okay, and WX is waxy. If it's waxy, it's puffy. If it's not waxy, it's wrinkled, okay? So little c should be yellow. Big C, I always put big C with a line underneath it, should be purple. Little WX is wrinkled. WRINK, sorry about that. And big WX is smooth. So when you perform this experiment, what you're going to be doing is looking at whether or not crossing over occurs during meiosis. So over here you have colorless or yellow and smooth, SM for smooth. Here you're going to have purple and waxy. I believe starchy might actually be wrinkled. No, that should be smooth. This is wrinkled. Dr. Val needs to look at her notes again. If crossing over occurs, you're going to see a recombination event between the colorless and starchy versus colored and waxy. When that happens, you're going to end up with a different combination. Now, again, using statistics, Mcclintock and her colleagues were able to predict whether or not you will see a recombination event occur. Now, based on this, we can do something called genetic mapping. I'm sorry, I'm going to cut that one short a little bit. It's a really fascinating story, but we're going to come back to the Native American corn when you talk about transpositional elements pretty soon. Now, moving on to genetic mapping. Genetic mapping is basically making a map of the genes along the length of a chromosome. So remember, you have a chromosome, and here would be the top, and here would be the bottom, and you're mapping from top to bottom. And you can do this by determining whether or not crossing over occurs. And this is what Morgan's group did. And so here are the Drosophila chromosomes. Here's chromosome 1, the X chromosome, chromosome 2, 3, 4, and then you also have the Y chromosome in there, which you don't need to worry about right now. So chromosome, the X and the Y chromosome is set number 1. Then you have 2 of 2, 2 of 3, and 2 of 4. So you have a total of 8 chromosomes in flies. We'll finish this setup in just a minute in the next lecture.